first talk today. And I'm going to be talking about carnassial tooth evolution and the macroevolutionary patterns within that, within the caniforms and the feliforms. So these are two suborders of carnivorous mammals. They diverged from one another around 60 million years ago and have since diversified into the many forms that we're familiar with. So within the caniformia, we obviously have dogs, we also have bears, weasels, raccoons and skunks. And then within the feliforms, you'll have obviously the cats, but then we also have the hyenas, the mongooses, and the civets and genets. As their name suggests, carnivora, the majority of these species eat at least some portion of their diet as meat. However, there's a lot of variability in diet within the carnivora, and in general, the evolutionary history of this clade is really one of repeated evolution into certain dietary ecomorphs. And one of the most famous of these really is the hypercarnivore ecomorph. So throughout the history of both the feliforms and the caniforms, we have this repeated evolution of vertebrate prey specialists. And I'm illustrating just a few of these here. And the people that study this the most are paleontologists. And when they're looking at the evolution of the hypercarnivores, they're characterizing this as evolution under strong functional constraint and morphological specialization. So you have increasing morphological specialization, increasing ecological specialization, leading to sort of very strong constraints on their morphology, taken to the extreme by these saber-tooth lineages, but in general, a lot of the various teeth and jaw morphology is thought to be highly constrained. And one of the characters that's consistently altered within all of these hypercarnivorous lineages is the carnassial teeth. So the carnassial teeth, these are, this is actually a modified lower molar, first molar, and a modified upper fourth premolar. And this is ancestral to all carnivores. And they've been modified so they form this really nice sort of shearing blade that allows the efficient processing of meat. And within the hypercarnivorous lineages, this has been modified and elongated. So that's what I'm illustrating here in these two lineages. We have the elongation of the teeth associated with hypercarnivory. So I'm going after a very sort of simple question here. The prediction that we're looking at is that we're expecting that the hypercarnivores will show much slower rates of morphological evolution in terms of the carnassial shape than the rest of carnivores, because what the expectation is that they're under strong functional constraint. But I'm also interested in whether we see differences between the caniforms and the feliforms. These two have been diverging, they diverged 60 million years ago, so there's been a long time for them to behave differently. And the expectation here is that we should see slower rates within the feliforms. Generally, they're thought to be better hypercarnivores and more constrained. So that would be the general expectation going in. To look at this, I have been looking at both the upper and the lower teeth and looking at the occlusal view. And I'm using geometric morphometrics and using landmarks. And I have 4 landmarks here. And ideally, you would like more. But the problem being within the feliforms, the cats, when they're hypercarnivorous, they have this very reduced morphology. They just have this big shearing blade. There's no crushing function. So it's harder to identify homologous landmarks between these two groups. However, we are able, with these 4 landmarks, to distinguish the 2 main sort of functional groups of this tooth, the slicing and the crushing portion. The trigonid is the slicing and the telonid being the crushing. So we measured and took this data on around 232 specimens, encompassing about 114 species, and that's members of every extant group of carnivora. We then combine that with diet estimated from the scientific literature, and we're using a measure that is very common within carnivore biology. So if you eat greater than 70% of your vertebrates in your diet, you're a hypercarnivore. We combine that with phylogenetic information to estimate multivariate rates of random motion, looking at the overall shape of the teeth. So I'm going to go into the results now, and I'm going to start with the upper tooth. So these are the results of the upper teeth. You'll notice here I have 4 categories. So we have only 114 species. We have a limited power with this data set. So I've stuck 4 categories. I'm looking at caniform hypercarnivores, filiform hypercarnivores, and I'm comparing those to the rest of the members of their group, sort of a general carnivore within the caniforms and filiforms. So we've got warm colours for hypercarnivores, cool colours for the general carnivores. So looking at this, you see 2 patterns. The first is that the hypercarnivores, both the orange and the red here, have significantly lower rates than the general caniforms and filiforms. And these are the post hoc p-values. When we focus in on the filiforms, we see that they're also evolving at a significantly lower rate than the hypercarnivorous caniforms. So these 2 results are exactly what we would expect given what has been previously published on carnivore evolution. But what's going on with the morphology? So here we've got the phylomorphospace that will help us understand the morphology. So each point here is a species, it's coloured for the category, and here the lines are the phylogeny. Then we've got PC1 and PC2 of our morphology. These explain over 80% of the total variance in our data set, and we can look here at the thin plate splines to see exactly how shape is changing. And if we look here at the hypercarnivores, we've got the red and the orange, they're clustering in morphospace. And we can see that PC1 is the one that is pulling them apart, making them distinct from the other carnivores. And so the morphology is one of elongation. That's exactly how it's been characterized. And the way they're doing this is they're narrowing the tooth, reducing the telonid, the crushing, and really increasing that trigonic, the shearing part of their tooth. You'll sort of be looking at this, you can see clusters of hypercarnivores. They're all in this region of morphospace, both the caniforms and the filiforms. This red cluster here is the true cats, the felids. So they're obviously more extreme and more elongate than the other groups. You'll probably also be wondering what this oddball is over here, and that's this guy, the polar bear. Bears are weird, and polar bears are even weirder. They are hypercarnivorous, but they have very omnivorous dentition. And it's debated whether either a very recent speciation event or there's been a high degree of hybridization with brown bears throughout their history. And both of those are likely to potentially prevent them from diverging morphologically very much. But in general, with the upper carnassials for the hypercarnivores, we're seeing exactly as we were expecting. Slower rates of morphological evolution, suggesting, or at least consistent with the idea of functional constraints, and we see lower rates within the filiforms. What about the lower carnassials? Do we see the same thing? And I'm going to give you the short answer, and that's no, we don't. We see something quite different. So when we're looking at the lower carnassial, if we just focus at the moment on filiforms, so we're looking at the red and the blue, we can see they have significantly lower rates of morphological evolution than the caniforms. So the 2 suborders here are behaving differently. But when we focus in on the hypercarnivores of both these groups, they have significantly higher rates of morphological evolution than their related general carnivore, which is certainly going against our strong expectations going into this study. So what's going on with the morphology? So this is, again, the phylomorphospace for those first 2 PC axes. The first thing you notice here is that both PC1 and PC2 are important for distinguishing the hypercarnivores. So what are these axes? So again, they're both elongation axes. It's just different ways of elongating the tooth. So PC1 is all about complete reduction of that telonid, the crushing portion. And we've got an increase in the trigonid, the shearing. Whereas PC2 is a narrowing of that tooth with the condylids coming into alignment. So we've got the caniforms and the filiforms behaving differently here. They're different clusters in the morphospace. And when we look at the filiforms, the only way that they're elongating their teeth and changing their tooth morphology here really is on PC1. So they're reducing the telonid and increasing the trigonid. Whereas in the caniforms, they're doing that to a certain degree, but then they're also narrowing the tooth. So they have two ways of getting towards an elongated tooth. You'll also, again, notice up here we have an outlier. This is the one hypercarnivorous mongoose that we have in our data set. And this also brings out another pattern. When we look here at the filiforms, so the red and the dark blue, you notice phylogenetic clusters in morphospace. So there appears to be stronger phylogenetic constraints within the filiforms. When you look at the caniforms, you've got lots of overlapping lines, suggesting probably greater evolutionary ability of this trait. And this is sort of confirmed when you look at the phylogenetic constraint. So just looking at phylogenetic signal within the caniforms and the filiforms for the shape of the upper and the lower tooth. So in both of these, the filiforms have a strong phylogenetic signal in their tooth morphology. Within the upper Carnassial, it's not that strong. Gradient motion expectation is 1, so we can see that it's neither of these have perfect phylogenetic signal, but the filiforms are higher. But when you look at the lower Carnassial, you see a really strong pattern here, that the filiforms are behaving very much like gradient motion, maybe even slightly more clustered than you would expect under gradient motion, suggesting really strong phylogenetic constraints on the lower tooth morphology, and very different from the caniforms. Okay, so that was a very quick overview of this, so what are we concluding? Well, I think one of the most interesting parts of this is that we see these different evolutionary patterns between the subunits of this integrated functional system, the carnassial teeth. They have to work together to be able to shear teeth, so your expectation would most likely be they should follow similar evolutionary patterns. However, we've got to also remember that these are two different types of teeth. We've got premolar and a molar, so potentially there may be differential developmental constraints acting here, allowing the morphology to be decoupled as well as even under the same sort of selection pressures. In particular, the thing that we found that really wasn't consistent with our prior expectation was that these hypercarnivores have much higher rates of morphology in the lower teeth. And if you were going to ask me why that is, I'm not sure. But I got a hand-wavy explanation that I need to go and see if our data is now consistent with. But potentially it could be a decoupling of the crushing and the shearing function. So within the lower teeth, the hypercarnivores no longer need a crushing part of their tooth. Potentially that's released them from any form of constraint on there, and so they're just reducing the back part of their teeth in any way possible, and that may be what's generating those faster rates. But I'm looking into that right now to see if that is actually what's happening. Related to this, we get that the hypercarnivorous cats and dogs have similar teeth morphologies in the carnassials in the upper portion of their mouth. They're both elongating their teeth in the same way, but in the lower it's different. And then finally, in general, we get filiforms evolving at lower rates. So I just want to end to thank you all for coming. I know it's difficult to get back from lunch. And to particularly thank Alec, who's one of my co-authors. He's been an undergrad working with me for the last couple of years, and without him, we couldn't do this work. He's doing all the geometric mathematics.